Well, welcome back everybody to the Cerebral Blood Flow virtual seminar series. This is actually our very last seminar for this series in 2020. So first of all, I'd really like to thank everybody for their participation in the series that we've been going. It's been going since July. This is actually all of the presenters we've had since July. So thank you so much to all of you for your great presentations and your engagement in this whole seminar series. And of course, to all of the audience as well. Thanks so much for joining us every other week for these last few months. I think this has been an outstanding experience for all of us. And thank you very much for your support. Just in terms of some stats, we've had 12 sessions since July, 12 keynote speakers, and also an excellent opportunity for 48 presentations from early career investigators and trainees. So thank you all for your participation in the series. We've had about 197 people register for every session, and we've got 771 unique email addresses now on our master registration list. So we've had a really outstanding reach for this seminar series across all continents. It's been really exciting to be involved in this whole series. We've had about 117 individuals attend on average every session. So about 60% of the registrants actually attend. And we also have all of our sessions on YouTube now. So we've had about 172 views per session. So all of you are really extending your reach beyond just the audience that attends these sessions. And so I think it's excellent to have those videos available for everyone who can't attend from different parts of the world. So thanks so much for your involvement and agreeing to have those videos posted on YouTube. Just as a note also, as we do every time we have these sessions, I just want to highlight the Cerebral Autoregulation Research Network. Please, if you'd like to join, send me your CV and I'll add you to our mailing list. And just to note, there's no membership fee for joining this society. And we actually have our next conference coming up in April of next year. Obviously, with the situation that we're all faced with the pandemic, we've decided to go ahead and just have this as a virtual meeting in April. Our abstract deadline is on January 15th of 2021, and you can get further information on the abstract submission process on the CARNET website. In terms of rules of engagement for the session, as always, please keep your microphones on mute and video off throughout the session. There will be time for questions at the end of each presentation. And if you'd like to ask a question through the presentation, you can either use the raise your hand function and actually you can turn your mic on and ask the question, or you can type the question into the chat and then we will ask that question for you as the chairs. This session is currently being recorded and will be posted on YouTube within the week. So just keep an eye out for the email that I'll be sending in the next couple of days in regards to that video. All right, so moving on now to the actual seminar for today. So I'm really excited to introduce my good colleague and friend, Dr. Trevor A, who's a professor at Mount Royal University in Calgary, Canada. Trevor and I have known each other for about 10 years now. First met at UBC Okanagan about 10 years ago at an ultrasound summer school, and really struck up a friendship and that's persisted for these last 10 years. So Trevor's got a really exciting research program at Mount Royal where he's training undergraduate students about the process of doing research, and is really producing some high caliber work out of that lab. So it's really impressive that Trevor's been able to take his training in the basic sciences and now is a very well respected human integrative physiologist. So I'm excited for Trevor to present his work today. Together with 3 of his undergraduate students. So we've got Valerie, Anna Maria and Taylor, and then also a PhD student who Trevor is co-supervising out of Ireland, Jack Leacy. So excited for Trevor to present today his work on cerebral blood flow regulation during incremental ascent to high altitude, the effect of superimposed hypoxia and hypocapnia. Thanks, Trevor.
Thanks, Carolyn, for the lovely introduction. And I'll just share my slide now and get started. Let me know if all's well.
Yep,
can everyone see it okay?
Yep,
okay, great.
Thanks,
Carolyn. Thanks to Carolyn and Patrice for organizing this. What an incredible service to our community. And I love that picture of all the all of the pictures of all the people that are presented, because it shows just how vibrant, excuse me, how vibrant and healthy our community is despite the COVID lockdown. So thanks again, and look forward to, really thrilled that you asked us to put something together and highlight some of the work that we're doing in Calgary and Mount Royal. So Carolyn asked us to put a session title together too, and so I was sort of reaching to try and find a common thread between all of the talks that we're going to give. When Carolyn Patrice asked us to put this together, I sort of looked around the room and thought, well, who do we have with current projects? And so we've put together these 4 suckers who said yes. And I suppose the common thread might be something around metabolic signals in the regulation of brain blood flow. We'll talk a bit about methodological perspectives and interactions between metabolic signals. And so that was our best attempt at trying to weave together these 5 talks today. So what I want to present today, I'll start with, is a perspective on cerebral blood flow regulation during incremental ascent to high altitude. And really, you're all here for the pretty pictures. And so I don't blame you if you check out after the middle of the talk where I show you some pictures. And want to really focus in on some of the superimposed effects of hypoxia and hypocapnia together. We all know as principal investigators that we're nothing without our collaborators. And that's certainly true with me working at Mount Royal at a small university. We're nothing without our funding agencies, 2 of which on this list no longer exist anymore. That was my passive aggressive dig at the current government, and I know Jeff Dunn will understand. Our industry partners that helped a lot, particularly AD Instruments, who lent us some gear for some of these studies. And of course, we need research ethics boards to help out, excuse me. And of course, we need our participants as well to volunteer for some of these studies, some of which feel invasive at times. And of course, we need our trainees as well. So a big shout out to the trainees that helped out with this work, and I'll highlight those in a second. And again, thanks to Carolyn and Patrice and the Cerebral Autoregulation Network. And wondering why it's just autoregulation and not cerebrovascular activity and neurovascular coupling as well, but we can talk about that later. So for those of you that don't know me or don't know Calgary, Calgary, Alberta, Canada is situated in the foothills of the Canadian Rockies. We're about an hour's drive away from the Front Range of the Rockies. We're a city of about 1.3 million people. Down in the bottom left there is the Bull River, and my apartment is actually not too far from downtown there. And it's a pleasure to live so close to the mountains, and those mountains have inspired me in terms of my own work for many, many years now. I work at Mount Royal University, as Carolyn said. Mount Royal is a small undergraduate-focused, teaching-focused university where we rolled out baccalaureate degrees about a decade ago now, but we are over 110 years old as an institution. So it's been exciting to be a part of developing undergraduate degrees and developing a research program in this context. And that whole research initiative and endeavor is quite new at our university. And so I'm really pleased to have been able to bring in federal funding and train undergraduate students in this context. And so I guess I created and run the Mount Royal University Integrative Physiology Lab, which is a combined teaching and research laboratory where there's a pretty high throughput of undergraduate students through 3rd and 4th year physiology courses, as well as in my research program. And here's just a picture of us getting ready for the 2017 expedition to Mount Everest Base Camp that I'll show you some pictures of today. In fact, there's Jack Leacy in the back there running the computer, and we actually just met when this picture had been taken. And so, my research work spans a lot of different topics. My initial training was in comparative animal physiology at the University of Calgary. My sort of first love is chemoreflex control of breathing, and with the influence of Phil Ainsley years ago, got into some high altitude hypoxia. About a third of my research program focuses on cerebral blood flow regulation, and I'll make it seem like that's a major topic today and that I know what I'm talking about. And lastly, getting into the integration of reflexes. So we've done some work on heart rate variability, breath holding, central sleep apnea, and more recently work around renal acid-base responses to counter the respiratory perturbations that you see at high altitude. And I should just do a shout out to undergraduate students too. I know it's rare to run a program primarily with undergraduate students, but the ones that want these research experiences are bright, they're motivated, and they work really well in teams. And so it's a pleasure to give them opportunities that they wouldn't normally have at bigger research-focused universities. My fingers are in many pies because of the kind of research program I run. But generally speaking, I'm interested in how the heart, lungs, brain, and kidneys talk to each other in response to stressors. And one of my favorite stressors these days is high altitude hypoxia and some of the integrated responses to acute and chronic exposure to high altitude. And so today, this is just a picture of us in the lab. Oh, my patient supervisor, Richard Wilson, just entered the waiting room, so I'll have the pleasure of admitting him. Hi, Richard. This is a picture of the lab. And I want to focus today a little bit about high altitude hypoxia and acclimatization, as well as cerebrovascular reactivity, perhaps in a non-traditional way of looking at it. We use rebreathing as a technique in the lab all the time. During rebreathing, where you're not able to clear the accumulation of chemostimuli, you see over time a drop in oxygen and an increase in carbon dioxide. And so these two chemostimuli accumulate together at the metabolic rate and can elicit both respiratory and cerebrovascular responses. We've also dabbled in breath holding as well. Craig Steinbeck, this is his fault for getting me into some of this work. And during breath holding, there are some differences between rebreathing and breath holding, not the least of which is the closed glottis and the sympathetic response elicited from that. But in terms of blood gases, it's the same kinds of responses. So these respiratory chemostimuli are similar between the both. And I remember asking Anthony Bain at the Hypoxia Symposium years ago when they were doing work on the elite breath hold divers in Croatia, how we might quantify the cerebrovascular responses against the accumulating chemostimuli during breath holding. And it was a sincere question at the time, and we spent some time thinking about it and ended up doing some work with Craig Steinbeck on this after thinking about it from there. So this is just work published from one of my first trainees in the lab, Rachel Scow, who's now working in the University of Alberta and just about to finish her PhD. And Christina McKay and I and the group wrote an education paper as well. So you can see here in the top left that you can see during rebreathing, this is the switch on to rebreathing. You can see that carbon dioxide, this is hyperoxic rebreathing, so we'll just discount the effects of oxygen when you're at the hyperoxic range. And so this is the increase in CO2 from a metabolic accumulation. You can see both the middle cerebral artery and the posterior cerebral artery are increasing in their velocity as well. And we take those as indices of flow. And you can even just see the slopes are steeper in MCA versus PCA. So we were quite interested in regional differences between them all. And in this case here, in absolute terms, of course, the starting point is different, right? The MCA has a larger resting velocity than the PCA. And you can see that the MCA has a steeper slope than the PCA. But I'm just going to move a couple of things out of the way on the screen. But you can see here on the right-hand side that when you normalize to the differential baselines, that difference in slope disappears. And so when you quantify a group of people, the MCA is almost double in terms of its responsiveness to changes in carbon dioxide compared to when you normalize them to baseline. And so it really matters how you present these data in terms of what conclusions you might draw with respect to the reactivity of differential cerebral circulations. So the CVR, the cerebrovascular reactivity to CO2, is larger in the anterior than the posterior, sort of, depending on how you look at it. And so with those perspectives in mind, just trying to advance my slides here. We published a paper with Craig Steinbeck's group, where we did some breath holding work as a part of a big team data collection in the lab in Edmonton at Craig Steinbeck's lab. And we wanted to develop a unique way, a novel way of quantifying cerebrovascular reactivity during voluntary breath holding. So this is just a schematic, raw tracing of participants when they're rebreathing. So you can see at the onset of rebreathing where this line is, you can see that you eventually develop a respiratory response, which is not the topic of today's conversation. You can see this drop in oxygen across the rebreathe and this increase in CO2, and those are driven by the metabolic rate, of course. During breath holding, of course, you can't, by definition, measure respiratory gases because they're not breathing. And so we thought about interpolating some of these data. So you can see here the end tidal oxygen starting point, and then the end tidal oxygen ending point. You can see over here the end tidal CO2 starting point and the end tidal CO2 ending point. And you can see you can interpolate these same sorts of responses. And so we wanted to compare the measured chemostimuli within subject during rebreathing compared to the interpolated chemostimuli during breath holding within the same subjects or participants. And so you can see here on the x-axis, we've got the measured end tidal CO2 during rebreathing and the interpolated end tidal CO2 during breath holding. And you can see they're right on top of each other, and that R squared was over 0.99. With respect to oxygen, on the x-axis here is the measured end tidal oxygen during rebreathing. On the y-axis is the interpolated end tidal oxygen during breath holding. And again, they're right on top of each other. But lastly, because those chemostimuli change together at the metabolic rate, we thought it was appropriate to try and find a way to quantify cerebrovascular reactivity to both chemostimuli at the same time. So we developed what we called a stimulus index. Because we know that cerebral vascular responsiveness is linear and positively correlated to increases in CO2 and inversely correlated to changes in oxygen, we set up the stimulus index to be CO2 over O2. And when we quantify the stimulus index across rebreathing versus breath holding, they're also right on top of each other. And so then we quantify the cerebral vascular reactivity in the anterior and posterior cerebral circulations using that stimulus index. And this is just a raw plot of one particular participant. You can see here in the MCA, you get that increase in slope, which looks an awful lot like the CO2 responses we talked about earlier. Every point here is a single measurement. And that's one of the strengths of the rebreathing technique that initially Reed and then Duffin developed years ago. And then here in the posterior cerebral artery, you can see that it's a blunter slope. And so you get similar kinds of responses that you get to CO2 rebreathing when you index the MCA versus the PCA against the stimulus index during voluntary breath hold. And when we quantified it, you can see that the MCA is larger than the PCA, much like we've seen before. So given this new development of the stimulus index, we wanted to apply this to other contexts where you might find changing chemostimuli at the same time, or conflicting chemostimuli. So these laboratory experiments, as we all well know, allow for well-controlled and isolated perturbations. But for those of us interested in field work, in these sort of so-called real-world stressors, CO2 and O2 chemostimuli are changing simultaneously. So it's difficult to control one variable and measure against another when you're in these sort of fieldwork contexts. And so often, as many of you know, again, we do these sort of transient blood gas perturbations to elicit various cerebrovascular responses. But a lot of this requires pretty heavy, technically difficult or heavy physically tech that's difficult to get to high altitude laboratories. Of course, Phil Ainsley and others have done a pretty good job of managing some of the logistics of that, but they're not trivial. Even just getting a calibration gas tank to a developing country to a high altitude location is very difficult. And so we thought that perhaps it might be interesting to look at just the steady state responses, that these steady state responses, that is to say, what is your cerebral blood flow now, given the prevailing chemostimuli now? And that steady state strategy to meet metabolic demands might be interesting to look at. So just as we'll just start at the beginning to assess a bit about what the problem is with high altitude stress. This is from a little book chapter I wrote for the textbook company years ago. You can see here on the x-axis is altitude in kilometers. On the y-axis is changes in gas pressures. The total atmospheric pressure here is in dark gray diamonds. And you can see that as you go from sea level in Vancouver, and then you ascend at altitude, that you get a drop in atmospheric pressure, and we all know this to be true. And then 21% of less is less, of course, so the PO2 also drops along the same way. Just to give you some comparisons, here in Vancouver, which is the closest sea level that we've got here in Calgary, it's about a 10-hour drive or a 1-hour flight away. You can see here that we've got about 760 and about 160 or so for P atmosphere versus PO2. Here in Calgary, we're just over 1,000 meters, depending on where you are in the city. So between 1,000 and 1,100 meters, you can see there's a drop in P atmosphere and PO2. Airplane cabins, back when we used to fly everywhere, are pressurized to a simulated altitude of 2,400 meters. And then lastly, way up at the summit of Everest, just below 9,000 meters, you can see that there's about a third of the available oxygen of sea level. So that's why this is a physiological stressor with high altitude descent. Coincidentally, about half the available oxygen of sea level is about 5,500 meters or so, which just happens to be close to Everest Base Camp, where I'll take you today. So in the laboratory, when we do acute perturbations, and this is adapted from Mark Poulin's lab, and Craig was there as a research assistant at the time when we first met. In fact, I might have been a participant in this particular study. You can see using end-tidal forcing when they start people at baseline gases in Calgary, which is a little below 40 for end-tidal O2, CO2, and a little bit below 100 for end-tidal O2. They drop them for a 20-minute hypoxic step. And you can see in the isocapnic case, you get a very large hypoxic ventilatory response. But in the case of Hypocapnia, when you let the CO2 float, you can see that you drop your CO2. So this robust hypoxic ventilatory response that elicited at initial exposure to acute hypoxia drops the CO2 in a poikilocapnic hypoxic condition. And so now you've got both hypoxia and hypocapnia, and those are conflicting chemostimuli with respect to the cerebral vasculature. This is from a great textbook chapter that Ainsley and others wrote, and you can see here that over time, with weeks of exposure to high altitude, you get this ventilatory acclimatization. So you get this concomitant hypocapnia that develops over time. So you're initially hypocapnic when you ascend, and you become more hypocapnic the longer you spend at altitude because of this ventilatory acclimatization, adding to this particular problem of these conflicting chemostimuli acting on the cerebral vasculature. This is the alveolar gas equation that you're all familiar with, but just as a reminder to you that changes in alveolar ventilation then affect blood gases in important ways. And so we'll just focus here on the blue line that you can see as you increase alveolar ventilation that your CO2 drops concomitantly. And we call this, with high amounts of alveolar ventilation, this is the metabolic hyperbola, that you get acute respiratory alkalosis because of this hypocapnia. So my first foray into high altitude physiology came from a very generous invitation from Phil Ainsley in 2012 or early 2011 to come on an expedition to the Italian Pyramid Lab. This Pyramid Lab was, I think, built in 1990 or so by the Italians, and is located just over 5,000 meters and just around the corner from Everest Base Camp. And you can see here the Mount Tomori, which is over 7,000 meters, and just across the valley from Everest is this pyramid shaped and inspired the architecture of this Pyramid Lab. And we spent 3 fateful weeks after about a 9 or 10 day ascent in this lab back in 2012. And I'm grateful for Phil and everyone else on that expedition. That really opened the door for me to start thinking about organizing my own expeditions, including Chris Willey, Kurt Smith, Sam Lucas, Ryan Hoyland was there as an undergrad for the first time and has become a bit of a phenom in the cerebral blood flow arena. In fact, he's on the line to answer the mechanistic questions for me later. So thanks to everyone for their organizational efforts and invitation. Oh yeah, and Mike Timko came along with us here as well as an undergraduate student, one of my first trainees. So it was a really important developmental experience for me. And the first time we started playing with incremental ascent and cerebral blood flow was this study we published in the Journal of Applied Physiology, led by Chris Willey, where we measured cerebral blood flow in the steady state during incremental ascent, the Pyramid Lab. Here's Chris Willey making measurements of one participant. Here's Kurt Smith picking up his cocktail on the floor. There's Phil Ainsley suffering from one of his many famous high altitude headaches. And we published the study just looking at basic steady state cerebral blood flow during ascent to 5,000 meters. So just to show you some of these data, you can see here at the top that when you plot both CO2 and peripheral oxygen saturation together, you get this drop in end-tidal CO2, which you'd expect because of the hypoxic ventilatory response. Because of that, oxygen saturation is relatively well protected until we get to about 5,000 meters when we go off a cliff. And then you can, when you look at cerebral blood flow, this is just global cerebral blood flow, looking at the ICA and VA, the internal carotid and vertebral arteries together. You can see this increase in cerebral blood flow across the entire expedition. But I just want to focus here on this incremental ascent model where we started in Kathmandu at about 1,400 meters, worked our way up to Namche at 3,400 meters, and up to Farache, which is just over 4,000 meters. You can see this increase in cerebral blood flow in relationship to these drops in these 2 different chemostimuli, which of course are antagonistic to bring blood flow. I mean, it looks like hypoxia wins here, yeah. So when we correlated every single SpO2 measurement during incremental ascent on every participant, you can see that there's a relatively good moderate correlation between drops in oxygen and increases in global cerebral blood flow. But of course, that didn't take into account those changes in CO2 that I just mentioned. And so that's been nagging at me for a long time, particularly when thinking about breath holding or other stimuli like this when gases change at the same time. And then also, what about cerebral oxygenation in the context of high altitude descent? So what we're going to set out to do today is to quantify an index of cerebral blood flow against both changes in CO2 and O2 in the steady state. And we want to develop this novel metric that we're going to call the steady state cerebrovascular reactivity. So this is not in response to an acute perturbation, the type we would do in a laboratory setting. This is a steady state change in oxygen and CO2 that you get whilst at high altitude. And we're going to go back to the Nepal Himalaya to do this. So we've organized, we tried in 2015 and came very close. And a week before our departure, that 7.8 magnitude earthquake happened in Nepal that killed 19 people at Everest Base Camp and probably at least over 9,000 people in the Kathmandu Valley. They didn't need tourism, they needed aid. And so we canceled our expedition in 2015. And through a lot of resilience, particularly fighting with risk service departments, we were able to go back in 2016, where we took this picture at Trekker's Rock at Everest Base Camp. And then have since gone back in 2017, and I'll show you data for that from that expedition today, and again in 2018. And we were going to go this year, and we were thwarted by COVID. So a couple of times now in the last 5 years, we've had a difficult time going to Nepal, but we're planning on going back in 2022 if we can. And I know many other people have been affected by COVID in terms of travel as well. The Everest Base Camp Trek is a tent city that emerges at about 5,300 meters in the Sagarmatha National Park in Nepal. It is the end of the base camp trek and the beginning of the Everest climb. It's about 2,500 meters ascent, about 65 kilometers over about 10 days, depending on how fast you want to do it. You first land in Lukla at 2,800 meters and spend 9 or 10 days ascending to 5,300 meters. And so in 2017 was the largest expedition that I've co-organized. We co-organized this with Syracuse University, and we had people from University College Cork. Jack Leacy is here today as well, and will present data from a different expedition, but that was his first high altitude expedition with us. Investigators from the University of Alberta, who helped with a lot with the measurements I'll show you today. Jeremy Walsh and a team from Queen's University, and then a bunch of people from Mount Royal. So 31 participants, 12 Mount Royal trainees, many co-investigators and clinicians and trainees from 5 different universities. And we did an incremental ascent profile from Kathmandu and 1,400 meters to Fariche at just over 4,000 meters over 7 days. So I'll just depart from the setup and show you some pictures and then get back to the science. This picture was taken in 2018 as we ascended and flew to the Lukla airport, considered the most dangerous airport in the world, not by statistics, but certainly by consequence. In fact, there was a crash here. I believe it was in 2018, just on the edge. So pilots have to miss this ledge. It's about a 500 meter landing strip and about a 12% grade. And they have to sort of navigate this in the mountains and land in this spot. So pretty thrilling place to land. Some of our team were in a helicopter and followed the plane in and took this picture. From this location here at the end where there's a fence, there's a picture taken by our photographer, Ken Emery. So there's the landing at Fariche, in Lukla at 2,800 meters. And then you can see the tower just over here. Here you can see the team getting ready. There's Jack Leacy, who's going to present today in the back. There's Tom Broussard, who co-organized, and the big team about to start the Everest Base Camp Trek. This is the beginning and the end of the trek here in Lukla. It's a beautiful place to be, and I look forward to going back sometime soon. So just some beautiful pictures of our trek. There's the famous Ahmad Blam in the background. This is one of about 9 suspension bridges that you cross on the first 2 days of the Everest Base Camp Trek on your way up to Namche. Here we are in Namche. This is actually on the way down, but we'll pretend it was on the way up. There's Jeremy, which is big in grin there. What time it was to spend all this time in the mountains with our Sherpa team and investigators from all over the world. These mountains look big here, my students always say, but they haven't seen anything yet because it gets bigger still. So there's Namche on day 3 at 3,400 meters, where we did our first set of measurements in basically lodged bedrooms, like the one I showed you with Chris and Kurt earlier. This is a picture from above Namche. And whenever I look at this picture, it just makes me, I'm bewildered that we get to do this for a living. And then up we go to Fariche. And so here's the team ready to ascend even further after our measurements in Fariche, where I'll show you data in a second. There's Marina James, who's now a qualified ultrasonographer in clinical settings at Edmonton, but she did all the cerebral blood flow measurements for us from Craig Steinbeck's team that we'll show you in a second. And then there we are at the Pyramid Lab. It was quite emotional to be back at the lab after so many years, after spending 3 weeks with Ainsley's group there in 2012. So it was a real pleasure to see it again. And just around the corner is Everest Base Camp, just past a village called Gorak Shep. So there's the team in 2017 making it to Everest Base Camp, and people look mostly happy, except for poor Garrick here. And so you can see here this little glacier. This is the glacier that broke off, or part of that glacier broke off in the 2015 earthquake that brought an avalanche down across Everest Base Camp in this exact location that we're standing here. So then the next morning, just to kind of finish the story of our wonderful expedition, we wanted to work our way up to the summit of Kalapatar here. which is about 5,600 meters, the highest I've ever been personally. So we start before sunrise, and we've got our headlamps on, working our way up and watch the sunrise out over Mount Everest here. And then that's the view on the way up. I'll just take a beat. On the left-hand side, you can see the glacier ends. That's Everest Base Camp. And then the ice falls begin and takes a right-hand turn and up. And you can see there's the Nuptse Wall, Lhotse faces in behind, and there's the summit of Everest in behind as well. This is a picture taken at the summit of Kalapatar. It's a special picture for me because Tom Brutson and I co-organized this trip together and we made it. This is my partner, Jamie, who's a physician who came to help on that expedition. So it's a pretty sweet picture of us at the summit of Kalapatar with Everest in behind. And there's the view as the day gets brighter. You can really see the ice falls there, the beginning of the Everest climb. So really special place to go and see, and feasible if anyone wants to come. You don't have to be a climber to do this expedition, happily. So back to our regularly scheduled program and to the science. One of the many studies we did in this expedition was to investigate cerebral blood flow regulation and cerebral oxygenation during incremental ascent to 4,000 meters. We brought along a duplex ultrasound machine for volumetric flow of the ICA and the VA. We brought along a phenometer as well, which is an absolute pain in the ass to bring along in these expeditions, in part because of the power sources. We did some portable arterial blood gas electrolyte measures, and we brought along a portable Mears machine for cerebral oxygenation as well. And we aimed to characterize the integrated steady state cerebrovascular response to incremental ascent over 7 days to 4,000 meters, and to really characterize this novel metric of steady state cerebrovascular reactivity. We hypothesized that this novel metric that came out of that breath holding study we talked about earlier will better track the cerebral vascular response when we index cerebral blood flow against the concomitant changes in CO2 and O2, that those regional differences we've shown before would persist with altitude ascent, and lastly, that these cerebral blood flow responses would be related to cerebral oxygenation in the context of hypoxia. And so if you wanted to look this reference up, the data I'm going to show you now comes from this, this publication that we got out in the European Journal of Applied Physiology in 2019, and led by my student at the time, Haley Lefebvre, and her partner, Shaylee Zubelis, and we did a lot of work together on acid-base balance with respect to altitude ascent as well. Haley and Shay have since gotten into medical school at the University of Calgary. And we consulted with Ryan Hoyland on this for his expertise. So thanks, Ryan, and hopefully you'll stay on deck. So first of all, we made some portable blood gas measurements using these iStat machines. Some of you may have experience with these. They're a lot more portable than the radiometer units that many people use. They're cartridge-based, and so you can buy the cartridges that you want for the particular blood assays you want to run. And so here is Heidi Neiston, who's a registered respiratory therapist who did the arterial blood draws for us. In fact, she did, I think it was 14 arterial blood draws in the dining room at the Goretz Shep dining room at 5,200 meters. So pretty difficult to get participant buy-in, but we pulled it off and published those in Shay Zubelis' paper in JPhys about acid-base balance, if you're interested in that. And here's Tom Brutzart working in an idealized laboratory situation, running the gases for us. So just in terms of the data then, you can see here on the left-hand side is altitude, and the top x-axis is day 0 in Calgary at just over 1,000 meters, day 3 at 3,400 meters, day 7 at 4,200 meters during incremental ascent. On the y-axis, this is the PA CO2, and as expected, you know, some people hyperventilate a little bit during an arterial blood gas poke, but on average, we tend to get 34 or so in Calgary. You can see that there's a decline in CO2 with a hypoxia-compensatory response and acclimatization with ascent. The star means different than baseline from now on. The cross means different than the previous altitude. So incremental decline there with ascent as expected. And of course, that's driven by the hypoxia-compensatory response. That's driven by changes in oxygen. You can see oxygen saturation here, normal values in Calgary, and well protected by day 7 as well. There isn't an additional decline in oxygen saturation, largely because of the hypoxia-compensatory response protecting it. But when we set up this stimulus index we talked about earlier, which is the relationship between CO2 over oxygen saturation, because both of those responses are linear with respect to both ventilation and cerebral blood flow. And you can see that there isn't a change in these, but there's an awful lot of variability, and that might drive differential responses within subject. So we then did perform duplex ultrasound in Calgary as well as in Namche and in Fariche by Marina James, the ultrasonographer. I should thank, I think it's either either Kurt or Ryan who gave me the slide to borrow. So thanks for that. You can see here on the top is internal carotid artery, both the diameter as well as the velocity signal, and the vertebral artery for posterior, both the diameter and the Doppler signal. We then measure cross-sectional area and then calculate cerebral blood flow, so volumetric flow in mils per minute, and then we can calculate global cerebral blood flow as well, which is really just 2 times the ICA plus 2 times the vertebral artery. So what did we find? So as expected, much similar to Chris Willey's study in the Journal of Applied Physiology, you can see this increase in cerebral blood flow with ascent, that in Calgary versus Namche versus Fariche. You can see the ICA is a larger absolute brain blood flow in the ICA versus VA, not unexpected. And then global, you can see here about 600 or 700. And then with ascent, it's not significantly higher in Namche, but is significantly higher in Fariche. But when, and then when you calculate conductance and take into the influence of blood pressure, you can see that there's a little more of an incremental increase, that this cross emerges as different than the previous altitude. But now we wanted to index these responses against the stimulus index that we talked about before. When you do that, you get more of an incremental change, that all of these crosses emerge as different from Namche. So it looks like we better reflect the cerebrovascular response during ascent. And again, this is much like calculating a hypoxic ventilatory response, where you index the change in brain blood flow against the changes in blood gases. And lastly, you can see the steady state cerebrovascular conductance reactivity here as well. So you can see you highlight these incremental increases with ascent. And so it looks to be that these metrics seem to hold some water in terms of what they're tracking with incremental ascent altitude. The other thing too is because when we quantify the hypoxic ventilatory response, a change in ventilation against a change in gases, we see plasticity over time with high altitude descent. Perhaps this might be the most controversial thing I'll say, and I don't know that we have the evidence for it necessarily from a mechanistic standpoint, but this may suggest plasticity in the cerebrovascular response because there's an increase in these responses relative to the changes in blood gases. So what about regional differences? Perhaps you wonder why we care about regional differences. Early on, most people would just take measurements of anterior, given that the internal carotids and the middle cerebrals together contribute about 70% of cerebral blood flow. But those of us that are interested in the control of breathing might be interested in a different cerebrovascular territory than just the anterior structures. This here on the right-hand side is a picture from the 1950s from Mitchell's group, who discovered those central chemoreceptive neurons of the ventral surface. You can see here the vertebral arteries merging together to form the basilar. This is the ventral surface of a brainstem of a cat. And that's where these were some of many known central respiratory chemoreceptor neurons are found. And so, of course, if you want to learn a bit more about the integration between cerebral blood flow and respiratory chemoreflexes, then Ainsley and Duffin have written an excellent review on that. I think it was Dempsey's group wrote a paper in 2009 in JAP where they were tracking changes in brain blood flow with indomethacin, and then Hoyland, Ainsley, and I in 2015 in JPhys tried to follow up on that work. So there's a relationship between the control of breathing and cerebral vascular regulation with respect to CO2 washout from these central chemoreceptors. And that's why this is one of many reasons why we might be interested in regional differences between cerebral blood flow. So Chris Willey first pioneered some of this regional difference work in 2012. You can see here in absolute terms, this is in volumetric flow or in cerebral blood velocity. You can see this relatively large increase in the internal carotid versus the vertebral artery, or the MCA versus the PCA. And when you quantify it in absolute terms, it looks like the ICA, that is to say the anterior cerebral vasculature, has a much larger response than the vertebral artery or posterior. Problem is, and Val Cates will talk more about this methodological perspective in a minute, when you quantify things with respect to normalizing to baseline, where the posterior vessels have a smaller baseline, you get a relatively larger response in a percent change difference. So the answer to the question, which has a larger response to oxygen, really depends on how you quantify it and how you show the data. And so this was a bit messy, so we wanted to follow up on this and look at it a different way. So we're going to calculate deltas from Calgary to Faribault in day 7 and look at all of those metrics we previously reported with ascent in absolute terms and in relative terms. So you can see here the delta response in the ICA looks larger in terms of cerebral blood flow compared to the vertebral artery, but in relative or percent change terms, that difference goes away. What about this new metric of steady state cerebrovascular reactivity? When you calculate the change from Calgary to Faribault over 7 days, it's a much larger ICA response, internal carotid compared to vertebral. That difference goes away in relative terms. When you look at conductance, the delta conductance, a bigger ICA change compared to VA, that difference goes away in relative terms, and you're getting the point here. And lastly, when you look at conductance reactivity, that is to say indexing against our stimulus index, there's a very big regional difference between the two that goes away when you normalize it against baseline values. And so it looks to be that there's differential regional differences in these cerebrovascular responses during high altitude ascent that go away when you normalize it. So the question, of course, remains, how do we show our data? I'm not sure I have the answer for you. We can go around in circles talking about which is more appropriate. And Val Cates will spend more time on that coming up next. So what about oxygenation? I'll just quickly cover some of these relationships with oxygenation as well and wrap up. We used an INVOS NIRS system that a clinician from the University of Calgary brought along for us. And we only measured NIRS across the forehead for the anterior territory on the same ipsilateral side as the MCA, the ICA that we measured. So we can relate cerebral oxygenation to internal carotid artery blood flow. So here's arterial oxygen content during ascent. Here's Calgary, kind of normal values or so. A drop with ascent on day 3, not a further drop on day 7, largely because probably of the hypoxia response increase in oxygen saturation. There wasn't a significant increase in hemoglobin yet in day 7, but a slight increase perhaps that may have, with some variability between subjects, maintained oxygen content. And lastly, when you look at cerebral oxygenation, you can see that there is a drop that isn't further dropped in day 7, at least statistically. So you can see their cerebral oxygenation is lower with ascent as expected. So we wanted to correlate these new metrics. And so here's just ICA cerebral blood flow. On the ipsilateral side is the cerebral oximeter probe that we used. On the y-axis is cerebral oxygen saturation, relatively moderate correlation between the two, and perhaps that's not unexpected. And that to us tells us that this regional cerebral oxygen saturation is relatively it's reflecting something real. You can see when you correlate cerebral oxygen saturation against conductance, a similar kind of moderate correlation. But when you index cerebral blood flow against the SI, as we've done before, your correlation is much stronger, almost 0.8. And lastly, and this is just at 4,200 meters when people are most hypoxic, which is why we did them there. You can see against conductance reactivity against the SI, also a relatively strong correlation. And so perhaps quantifying cerebral blood flow against the stimulus index is a better way to go about assessing cerebral blood flow responses that are related to cerebral oxygenation. So what have we learned here today? Just to wrap up, from a methodological perspective, we developed this novel metric that we call steady state cerebrovascular reactivity, not in response to a transient laboratory test, but in response to steady state changes in O2 and CO2 together. We think that these capture the steady state cerebrovascular responses to these concomitant conflicting blood gas stimuli that have opposing effects on the cerebrovasculature. I think pretty conclusively, we've demonstrated regional differences in the absolute steady state changes in anterior versus posterior cerebrovascular responses during ascent. And lastly, it looks to be this novel metric where we index brain blood flow against the stimulus index was well correlated with cerebral oxygenation and hypoxia. So it looks as if I've reached the finish line on my talk today. And I'm happy to turn it over to Carolyn for questions and look forward to opening it up for our students to present as well. So with that, thanks for your attention, everyone. And I'm happy to take a few questions or at least pass them off to Ryan.
Thanks so much, Trevor. That was an excellent presentation. And I will apologize in advance for my dogs barking in the background because they're, a rowdy bunch as I ask the questions today. So I actually have a question for you, Trevor, just as people type in their questions into the chat. In regards to the cerebral oxygenation Were you surprised that there was a linear relationship between blood flow and cerebral oxygenation? We think about cerebral oxygenation being perhaps more protected due to increased extraction of oxygen from the blood. Could you just kind of speak to that finding that you have?
Sorry, which one are you talking about specifically, Karen? Are you talking about the linear? Sorry, just so I'm clear on what you're asking.
Just the NIRS data that you presented at the end there with the cerebral tissue oxygenation.
Yeah. This is outside of my wheelhouse, so I'll just wave my hands around first for that. But we were just exploring the data and noticed those linear relationships. And so I don't know what to say with respect to oxygen extraction. We did this at one altitude only. And so all those correlations showed was that those people that had larger brain blood flow responses against the prevailing chemostimuli also had better protected oxygen saturation in the brain at one altitude of 4,000 meters. So, Jeff, do you want to jump on and tell us your perspective on this? Because I expected to hear from you on this, actually. So,
yeah, certainly. I'll just show my face. So,
good to see you.
Yeah, good to see you. Nice talk. Always good to hear. So some years ago in the Hypoxia meeting, we were talking about whether or not brain oxygen would be preserved. So we did a study where we implanted PO2 probes in rats, and in a way that we could have the animals recover. So the animals were awake. And we recorded tissue PO2 as we dropped their inspired gas. And it drops linearly until about an inspired oxygen of about 10%. And between 10 and 8%, it starts to taper off and get pretty critical. So I think around 8% inspired oxygen in an awake mammal is where you're starting to get this critical delivery problem, which might relate to the death zone. But above that, hypoxia doesn't stimulate blood flow enough to maintain PO2.
Interesting. Yeah, I mean, this is cerebral oximetry, looking at probably a mix of venous and arterial hemoglobin saturation. So it's not PO2 in tissue, I suppose. But you can see even at 4,000 meters, some people had values that were near baseline in Calgary, 70% or so. But in this case here, we're only at 4,000 meters. Your 10% stimuli is probably more like between 5,000 and 6,000 meters. Yeah, so a higher respective altitude.
And the other problem is, I'm not sure about the NVOS precisely, but probably over half of that signals from non-brain. So it's basically arterial sats driven.
Yeah, I've heard that it might be confounded by skin blood flow in the forehead as well, and various machines are different in that regard. So I also look at these with caution as well.
Yeah, it's good though.
Thanks for showing up, Jeff. Nice to see you.
Good to see you.
Since I don't have any dogs at home, I will replace Caroline for the next few questions. So, personal question from Mike Timko. Since you have gotten to altitude many times now in Nepal, do you find the acclimatization process easier each time you go? And have you felt different from a health perspective each time you go?
That's a fun question. These are always more fun questions to answer than the science ones. So thanks, Mike. Mike came with us in 2012 to high altitude for the first time, and we both found out that we did quite well at altitude, despite being semi-obese men at the time. When I went in 2012, we were on Dimox on the way up. In 2016, when I went, we were also on Dimox, and I felt quite sick at 5,200 meters my first day there. And the next day in 2017, the next year, 2017 and 18, when we went back, we weren't on Dimox prophylactically, and I did fine. I did probably better or just as good. So I don't know what that mystery is, whether there's people have sometimes discussed the possibility of a learning effect or plasticity in these responses. But for some strange reason, I do really well at altitude, as long as I protect my hydration and, do a slow ascent profile. So I wouldn't say I felt different, but I just do well generally. Mike, you've done this as well. So have you had a different kind of experience going to different altitudes with different ascent profiles? Mike says he feels worse every time he goes. Well, I think in the Andes, you had a pretty bad chest infection, I heard. So that might have played into it as well. Thanks, Ben.
Okay. Another question from Dr. Ron Benrai. Thanks for the excellent talk. Have you compared the steady state metrics with the transient approach at sea level or Calgary level?
That's a great, thanks for showing up, Dr. Panerai. Great to hear from you. That's a good question. And so this was our first foray into the steady state responsiveness. I've since moved the steady state model into the respiratory responses with Ascent, and you can check out Jack Leacy's recent publication in Experimental Physiology, where we looked at steady state respiratory responses during Ascent. So I haven't compared the transient brain blood flow responses to these steady state responses, but I think they're kind of measuring different things. This is just the steady state strategy that you're sitting at given prevailing chemostimuli, but a perturbation, a response to perturbation, I think is a different kind of thing. So ultimately it comes down to, I think what your experimental question is in terms of what you're looking at. These transient chemostimuli may not be that relevant unless you're thinking about, say, sleep apnea when you have a transient breath hold. But sorry, I haven't done any of these kinds of comparisons to different kinds of stimuli in the lab. So it's something we should probably investigate in the future.
Great. I think that we have a few comments and questions in the chat. So maybe a little bit later if you want to answer, Trevor. So I will pass you the puck for your team now.
Yeah, great. I'll just say Ryan Hoyland is put in a perspective for NEARS. So thanks for that, Ryan. I'll let everyone read that. But I appreciate your expertise, Ryan. But I'd love to move to our students if we can. So thanks everyone for coming and listening. And I'll pass it back to, actually, I guess I'm taking it over now, aren't I? So I'll just put on this intro slide just to let everyone know with respect to what's to come. We're going to have Val Cates come on, who's an undergraduate student from Mount Royal University. Anna Maria I thought I had it, Anna Maria, sorry. Ciorogariu-Ivan, who's just finished her BSc from Calgary. Taylor Bader, who's just finished his BSc and started a master's. And then Jack Leacy. But we're going to swap Jack and Taylor, if you don't mind. The way we did these studies, we started with Jack's study and then did a follow-up that Taylor led. So we're just going to swap Jack and Taylor. Val, Anna Marie, and Taylor will give 10 or 11-minute talks, and Jack will give more like a 15-minute talk. So that's what's to come. So without further ado, I want to introduce Val Cates, a rock star undergraduate student from Mount Royal who's finishing her 4th year BSc in health science, who asked to get involved last summer and volunteered with some data analysis and some writing, and that's what she's going to present to us today. So I'm really excited for Val to have this opportunity and looking forward to your talk, Val. So go ahead and share your screen.
Well, thank you for introducing me, Dr. Day. As Trevor Day just said, my name is Valerie Cates, and I'm finishing up my undergrad at Mount Royal University. And this summer, my partner, Joanna Keogh, and I, who's first author there, did a little bit of research on some previously acquired data into regional blood flow responses during acute hypoxia within healthy humans and how we best report those response changes. So with that, I'll hop right into it. Just a few facts about the brain before we get going today. It's a pretty small organ compared to the rest of your body, but it takes up a large amount of your metabolic energy at rest. And unlike other organs, it cannot readily store nutrients or metabolic waste that accumulate. As such, it must rely on adequate blood perfusion to meet changing metabolic demands. And so in the case of increased metabolism, generally we're going to need more oxygen. We're also going to produce more carbon dioxide, both of which have a role in altering the vessel diameter. So in the case of low carbon dioxide, generally a hypocapnia, we see vasoconstrictory effects and increased resistance within the vessel. Conversely, with low oxygen hypoxic state, we see vasodilation, and this decreases resistance in the vessel. Because resistance is inversely proportional to flow, we can use changes in resistance or velocity measures as an indicator of blood flow through certain conduit vessels, given that they're not changing their diameter. What are the conduit vessels, you may be asking? The ones that my partner and I looked at were the middle cerebral artery, coded here in yellow. It's perfusing about 70% of your brain at a given time. And we also compared it against the posterior cerebral artery, coded in coral. That guy's perfusing about 30% of your brain at a given time. And so the way we do this, the way we measure changes within these vessels, we use a technique called transcranial Doppler ultrasound. It's relatively easy to access. All of us undergrads at some point have had a little bit experience playing around with it. And what it does is it measures blood velocity, so the speed at which red blood cells are coming towards or away from the probe. And because the velocity of those red blood cells is indicative of changes in resistance, and resistance is inverse to flow, we can use that as an approximation of changes in flow. As Trevor previously stated in his presentation, a lot of studies so far have well examined the effects regional responses to carbon dioxide on cerebral vasculature, but there have been fewer studies that have looked at influence of oxygen on those vessels. So another thing to consider besides the arteries we're looking at and the gas perturbations that we're choosing is the way we are actually reporting and quantifying those changes. So the way we do so really matters, as Trevor was alluding to. Essentially, we can do two styles. So we can do an absolute, which is just the net change of where you start and where you end, or we can do a relative change, which takes into account the starting points of where we begin. And so just to illustrate that with these two little people on the right-hand side of my screen, they're both starting out with a set number of coin. Person number 1 has 1, person 2 has 2 coins. If we were to give each individual an additional coin, the absolute change is the same for both individuals of +1, but the relative changes are very different because of where they're starting off. So going from 1 coin to 2 coins is a 100% increase, but going from 2 up to 3 is only a 50% increase. So even though the absolute change is the same, because they're starting at different points, the relative change is going to be different. So based on all that, the research that we did this summer, we wanted to determine how acute hypoxia alters cerebral blood flow to the anterior and posterior regions of the brain, looking at the MCA and PCA respectively. And then we also wanted to see how reporting in relative or absolute terms changed the findings from above. So what we hypothesized was that the anterior vasculature would be more responsive to that acute hypoxic perturbation than the posterior would be, and that this would only be observed in absolute terms and not relative, just simply due to those differences in starting baseline values, remembering that the middle cerebral artery perfuses about 70%, whereas the posterior is only 30%. So how this looked, the equipment that we used, transcranial Doppler ultrasound, as I previously described, just this fancy little headpiece, a little ultrasound probe that can detect the speed of those red blood vessels and the direction, allowing you to figure out which artery you're essentially looking at. We also used a phenometer to track changes in beat-to-beat blood pressure, MAP, mean arterial pressure, and then we used an end-tidal forcing system that was designed by Dr. Glenn Foster. And what this device does is it basically allows us to clamp and set the inspired gas measurements. And then there's a gas analyzer at the other end that will examine the amount of gas that the participant is breathing out. So locking what goes in and determining what's coming out. And so the protocol for this experiment, it was done on 14 healthy participants. They were all rested in sort of a supine position. And the first part was the baseline measurement, where they just breathed normoxic room air for about 5 minutes with set sort of predetermined carbon dioxide values for what was good and normal for those individuals. Following that, we did the experimental protocol, which involved clamping the inspired oxygen at about 45 millimeters of mercury or torr, leaving the carbon dioxide at their set normal value, and then also trying to get their saturation of oxygen down to about 80%. All of this to simulate a steady state isocapnic hypoxia, pardon me. And then following that, we just did a 5-minute recovery, which was almost exactly the same as baseline, allows for a washout of the gases that accumulated in the blood. And so what we found here can be kind of illustrated in this figure. I'll just walk you through this. This gray bar is indicating the hypoxic sort of experimental protocol element. We've got the baseline elements before and the 5 minutes of recovery afterwards. And so what we can see here, these little white dots are denoting the amount of expired carbon dioxide in the breath. Each dot is representative of all 14 participants, a 15-second bin, the average among all of them. And so what we see is the carbon dioxide that was expired was relatively constant at about 40 torr. Similarly, looking at the black dots is indicative of the oxygen that they were breathing out, oxygen content in the expired breath. That dropped down to about 45 torr. And then similarly, the saturation of oxygen also dropped to about 80. So all this is indicating is that we did reach a hypoxic state within an average of all the 14 participants. From there, what we did was we plotted the cerebral blood flow velocity of the MCA and the PCA. PCA here is in white, MCA is in black. What you want to notice is that in both cases during the hypoxic perturbation, there was an increase in cerebral blood flow velocity. And so with these little black bars, we basically took the sort of data points from those 2 minutes and subtracted 2 minutes of the baseline to get the absolute change. And we took the same points to do calculations for the relative changes. And so what we found when we plotted this all out in graphical format, in absolute terms, we found generally that the middle cerebral artery was more responsive to hypoxia than the posterior cerebral artery. What each of these little dots is indicating is just the participants' blood vessels. We've got this black bar denoting the average of all the participants and then the standard deviation. So in terms of the change, absolute change, the middle cerebral arteries showed an absolute change of about 17 centimeters per second, 17.5. Whereas the PCA was only about 11.2. And these two values were significantly different from one another. When we did the same sort of plotting with the relative changes, though, we saw that they were almost identical to one another. So they were not significantly different. Again, each little dot is a participant's blood vessels, PCA, MCA, average of all of them, standard deviation. And so what's interesting here is when you do a relative calculation of these two vessels, they're almost exactly the same from one another. And so based on this, we kind of did a little bit of a meta-analysis to see what other literature is out there in terms of regional responses to hypoxia. When we looked into studies that reported the absolute changes, these were the ones that we could find, including ourselves here. There's a lot going on here, but what I just want you to bring your attention to is this red box, the cerebrovascular reactivity to hypoxia versus anterior versus posterior. In all cases, the studies we found that reported absolute changes consistently reported that the anterior was more responsive than the posterior. The same cannot be said for the relative changes. So again, same sort of style, just looking at studies that report relative changes. There's a handful that report posterior being more reactive than the anterior in response to hypoxia. There's even more studies, including ourselves, that found that they were very much equal to one another. And then even more studies illustrate that the anterior is actually more responsive to hypoxia than the posterior vasculature is. So what's interesting here is, even though there are so many more studies in relative terms, they're very inconsistent as to which region is more responsive to those changing blood gases. So that's really important for scientific transparency. If we're reporting both absolute and relative style, we can all kind of, as a scientific community, better interpret that data and use it to further our own research. Relative can be great for clinical settings because they reflect the sort of personal initial baseline values, but they're a little bit misleading when we start looking into a research setting. And so by just adding that addition of the absolute reporting, it can really help further the research. So just to summarize here, we confirmed our hypothesis that acute isocapnic hypoxia elicits regional blood flow changes between anterior and posterior vasculature, with the anterior being more responsive only in absolute terms and not relative. When we did our little meta-analysis here, the vast majority of them will report in relative terms, but it's very inconsistent as to which region of the brain vasculature is more responsive. And only a few studies reported absolute changes, but those that did were consistently finding the anterior was more responsive to hypoxia than posterior. And so with that, I just want to extend thanks to my research team this summer for letting me join on. I want to thank everyone who did the initial experimental design and collected all the data. And then special thanks to our hosts, Dr. Carolyn Rickards and Dr. Patrice Brassard, for organizing our event today. My email is just in the bottom corner if you can point me in the directions of any more studies to add to our meta-analysis. But with that, I will just open it up to questions for you guys.
We're all giving you jazz snaps, Valerie, so well done. Thank you so much.
Yeah.
So thanks for sort of rounding out our discussion about our absolute versus relative differences. Don't see anything in the chat box so far. Jill Barnes and I are just talking about politics privately, but otherwise.
I'm an undergrad, so I can't answer in the same way that Trevor can, but I will do my best. If there are questions.
No, I think probably better, Val, probably better. The imposter syndrome is real and it never goes away.
Sure is, yeah.
Lawrence Lebrec has asked, do you think considering biological sex could influence the results, either in absolute or relative terms?
I'm not too sure. I mean, some of the studies I did look into reported differences in sex here, so it could be. I know that, who was it? I think it was Morris, actually. Morris and her team, they specifically looked at women exclusively. And they also included children and adults, and they found differences within age differences. Sex differences could certainly be an area to explore. I think as we're all kind of learning in in my undergrad program, is that women are very underreported, or minority groups are not representative in scientific studies. So it would definitely be interesting to look a little bit more with a focus on sex differences. I'm not too sure if there is one, but I do know there are differences in age responses.
Mark Chertoff is asking a mathematical question about analysis, Val. And I think you and I haven't talked about this, but I'll just read his question. He's asking if we've looked at time constants as opposed to just the absolute change between baseline and the point later in the hypoxic response.
Yeah, I have not. My partner, Joanna Keogh, did a lot of our data analysis because she had sort of the previous experience taking Trevor's classes before. I'm not too sure if we have. Trevor, do you know at all?
Yeah, the short answer to Mark is no, we haven't. And so we might want to think about looking at, it's just a way of mathematically looking at the on response during hypoxia. Because we just looked, if you go back to the slide that shows the quantification, Val, we kept it relatively straightforward, Mark, in as much as the next one. Yeah, so you can see, the raw tracing. Yeah, you can see we just took a bin, I think it was a 2-minute bin near the end of achieving steady state. And so if we were to track the on response early on and look at the temporal dynamics, we might find differences as well. So no, we haven't, Mark, but thanks for the question. Minu Ashuri is asking, how did you create hypoxia in the participants?
Yeah, so essentially, like we weren't the ones who actually collected the data because this happened, this was all collected many years before COVID. If I understand right, based on their methods, essentially they did that end tidal forcing system. So they were able to basically control the amount of inspired oxygen that each participant was breathing in, and set it to a value that would be representative of a hypoxic state, which normal oxygen would be about 100 millimeters of mercury. So they dropped it down to about 45 torr, because that would be pretty indicative of a hypoxic state, and then also measuring those saturation levels to ensure that oxygen content within the blood was also dropping as well. So basically it's just a fancy way of clamping the inspired gas, in this case oxygen, to create a hypoxic state.
Jill Barnes has asked, and I think this is important to think about, and particularly because you've done a meta-analysis, Val, thank you for highlighting the left and right arteries. Do most of these studies evaluate both left and right vessels when examining anterior versus posterior?
Definitely, some do. The Kellowan one was quite comprehensive. They did a whole bunch of other vessels within the brain, and they were able to look at the right and the left using MRI essentially. All of these studies, I could definitely go through and elaborate on which ones they chose to look at between right and left for posterior and anterior. I don't have those numbers off the top of my head, but I'm fairly confident that they at least list it within their methodological outlines.
There's a few more questions that come down to some analytical differences that perhaps we'll just highlight and talk about later, Val. Austin Davis is asking us whether or not we've thought about baseline as a covariate. So we'll think about that with respect to statistical technique. And then Patrice has said, although there's no differences between MCA versus PCA in relative terms in response to hypoxia, there seems to be two distinct responses when looking at individual data. Can you explain these responses between subjects? Perhaps, Patrice, you can just highlight what you mean. Maybe go back to the raw tracing again, Val.
Oh, no, sorry, but you were on the right slide when you look at MCA versus PCA in relative terms.
Yeah.
You have like half of the subjects have like higher MCA versus PCA, and the other have the opposite.
Mm-hmm. Yeah. I mean, it seems like it's all over the place. Again, my partner Joanna did a lot of this analysis, so I'm kind of going off what she has filled me in on. But yeah, it does show each participant is going to be different in that regard. But I think when we took the averages, we kind of used that as our indicator of like, is there something significant between them? But yeah, it is interesting to note that some people actually decrease within anterior to posterior. Other people go the opposite way. So again, I think it would have been beneficial if we could actually conduct our research, like do the actual experiment and collect all the data ourselves. I think it would help me understand it, but as it is, I'm not too sure why they would be different between individuals specifically quite yet, but I can do more research, so.
No worries, and it highlights the importance of showing like individual data on top of what
exactly, yeah.
Thanks.
No problem.
I agree, Patrice. And the other thing too is just highlights the inherent variability in human studies as well. Like the variability is not the exception; it's the rule, and we have to take into account and recognize the variability. So thanks everyone for your questions. Val, well done, and thanks for your presentation.
Thanks, guys.
Perhaps you can stop sharing your screen, and Anna Maria is on deck.
Hello, everyone.
Hi, Anna Maria. Thanks for joining us. It's my pleasure to introduce our second undergraduate speaker for the event. This is Anna Maria Ciorogariu-Ivan. I tried.
You did a great job.
Thank you. Thanks for being nice about it. Anna Maria just completed her BSc in health science last spring and had the enviable experience of being able to do a drive-in graduation because of COVID. Anna Maria took my advanced physiology class last fall and wanted to do an independent project in the winter. And so, you never say no to someone as engaging as Anna Maria. And she, I think, breaks the record for number of participants collected before the COVID shutdown in March. So all of these data that you're going to see were collected between January and March, right before the university shut down because of COVID. So with that, Anna Maria, please go ahead.
So thank you for that introduction, Trevor. Like he said, my name is Anna Maria Ciorogariu-Ivan. He was very close, but just doesn't have the background to really roll those R's, I don't think. Like he mentioned, we worked from January to March to collect and analyze data for this kind of research. We were looking at what would happen to this brain blood flow regulatory mechanism called cerebrovascular reactivity, particularly in terms of carbon dioxide, and what would happen if we gave people, healthy participants, a lot of sugar all at once. So I think the natural starting place to begin talking would be to start talking about sugar. I don't think there's one person in this chat who's unfamiliar with having a sugar high or sugar rush. It's an important nutrient, and it's also incredibly easy to get, and it's super important for your brain, but we almost have too much of it. The daily recommended intake for men is 37 grams, and for women it's 25. And it's very easy to go over this limit. In fact, Stats Canada reported that in 2015, the average Canadian consumed 105 grams of sugar a day. This includes both fruit sugars and table sugar, so we go over quite a bit. your fasted blood glucose is about 5.6 millimoles per liter when you're just fasted. So you haven't eaten anything yet, you haven't had any of your morning coffee, you're just fasted. And if you do have something to eat, or if you have an oral glucose tolerance test, which I'll be talking about more later, it should show less than 7.8 millimoles per liter after 1 hour. This is called an acute hyperglycemia. Your blood sugar is slightly raised. And an acute hyperglycemia is important because this causes high levels of free radicals in the blood, and this can prevent nitric oxide from being available for target cells. So I've included a picture there on the right to show you which target cells I'm talking about. Of course, I'm talking about endothelial cells, which line your blood vessels. If you don't have nitric oxide available for these cells, they don't do their regular job, which would be to cause vasodilation, of course, expanding of these vessels. And that can cause a cascade of problems, starting with increased platelet aggregation and viscosity, and that increases your resistance and your blood pressure. So all in all, not a great thing. Nitric oxide is, of course, important for general systemic circulation, but it's also really important for cerebral blood flow. And of course, since your brain is highly perfused, this needs to be highly regulated as well. And one of the regulatory mechanisms that's used to monitor your brain blood flow is cerebrovascular activity. And this is in particular to arterial blood gases, so carbon dioxide, oxygen, and pH. These arterial blood gases regulate cerebral blood flow through endothelial-mediated nitric oxide. So we're seeing a little bit of a relationship there. Hypercapnia is high carbon dioxide. And we're talking about carbon dioxide because that is, of course, the variable that we're interested in. If you have high carbon dioxide levels in the brain, your blood vessels will vasodilate. And if you have too little or hypocapnia, you'll get a vasoconstriction. And that's due to cerebrovascular reactivity. Now, we know that some factors that can influence cerebrovascular reactivity are hypoxia, sex, age, exercise, sleep, and blood pressure changes. And these are, of course, in response to varying carbon dioxide levels. But it hasn't really been elucidated whether glucose is relevant to this as well. So we wanted to look at regulatory mechanisms that have already been demonstrated to be altered by acute hyperglycemia, and the other regulatory mechanisms being neurovascular coupling and cerebral autoregulation. Of course, my colleagues, Taylor Bader and Jack Leacy, will talk your ear off about neurovascular coupling, so don't worry about that. And nitric oxide levels, as previously mentioned, are altered by acute hyperglycemia, and this is important for the cerebrovascular reactivity to carbon dioxide. So the aim of this study was to characterize the effects of acute hyperglycemia on cerebrovascular reactivity to carbon dioxide, and we hypothesized that there would be a decreased response to carbon dioxide following acute glucose loading because of the aforementioned variables. So this participant on the right here is fully instrumented. She's in the middle of one of her trials, and she did give permission to be in this slideshow, although she didn't love the picture. We did collect data from 23 participants. All the participants came to the lab fasted for 12 hours. This meant no caffeine, no sugar, just water. And of course, for the safety of participants, we wanted to make sure that anyone with any cardiovascular, respiratory, or metabolic disorders would be excluded from the study. So in terms of instrumentation, one of the first instruments that we used was Acute. This was to check the levels of blood glucose before instrumentation, following an intervention, and we did that 4 times during the protocol. I use a little arrow here to point to that tubing coming off from the mouth. That's pointing to the end-tidal gas analysis to look at end-tidal CO2. This 6-liter rebreathing bag was filled with 7% carbon dioxide and 93% oxygen. And the switch just above that could switch from between room air for when you were doing baseline and switching to the hyperoxic rebreathing bag. And the headpiece there was connected to the transcranial Doppler ultrasound, measuring her middle cerebral artery for blood velocity changes. And a picture of the TrueTol, which we used for participants to increase their blood glucose levels. So just a rundown of the protocol. We did the blood glucose tests to begin with, making sure that they were fasted and in those normal ranges. Following that, we would instrument them and give them a 5-minute baseline where they would just breathe room air. Then we would flip the switch, so they would be breathing from that 7% carbon dioxide. And we would do that until they reached either an end tidal CO2 of about, I think it was maybe 38 or so, or until they just couldn't do it anymore. And then we would get the participant to drink from that Trutol. It was 75 grams of sugar and 300 milliliters. And then we would wait 30 minutes, check their blood glucose again, and rerun that entire protocol. So once we had their data, we analyzed everything using 80 Instruments lab chart and Microsoft Excel, and we used linear regression calculations to calculate the response slopes to measure the cerebrovascular reactivity. So I included this data table here to include some variables that were measured in their fasted and their glucose-loaded states. And I'm highlighting the ones that changed from fasted to glucose significantly, and that would be the blood glucose, the heart rate, the MCA velocity, and the cerebrovascular conductance, which is just MCA over MAP. I also included a raw tracing from one of the participants here. I have the middle cerebral velocity average on the y-axis plotted against the end tidal CO2 on the x-axis. And every single dot there is an analysis, beat-by-beat analysis of the participant's cerebrovascular reactivity. When we calculated the slope, we got the CVR, which was in this participant 1.89, and the linear, the R squared was a really good fit there. It was about 0.93. This was the blood glucose from every single participant in their fasted and their glucose states. As you can see, the individual dots was one participant, and the line connecting from their fasted to their glucose states. The bar in the middle there represents the average of all participants in that state. So in the fasted state, the average blood glucose was 4.7 millimoles per liter. And in the glucose state, the average blood glucose was 8.1 millimoles per liter, which is, of course, a very significant difference. And what you've all been waiting for, and I'm sure you'll all have a lot of questions about, is the CVR from all the participants. So this is the cerebrovascular reactivity from the fasted to the glucose states. Again, I included every single participant's average on here. And in the fasted state, the average CVR was 1.37 centimeters per second per torr. And in the glucose state, it was 1.04 centimeters per second per torr on average. So it did decrease, and this was a significant decrease of 0.005. So we're trying to make a conclusion that CVR to CO2 was reduced by acute glucose loading in healthy participants. There have been studies done on CVR being reduced in diabetic patients after acute glucose loading, but it's interesting to see that it can be reduced just from an acute hyperglycemia in healthy participants. And Trevor really wanted me to nail this point home, but we didn't have a clinical hyperglycemia in this study. Like I had mentioned before, our average hyperglycemia was 8.1 millimoles per liter. I think Mayo Clinic says that clinical hyperglycemia is about 10 to 11 millimoles per liter. So just having something super sugary could actually induce this lowered cerebrovascular response, which could be interesting for university students or any sort of student or anyone who needs to be kind of on the ball and might be sabotaging themselves by having like a sugary snack, thinking that they're getting their energy up. But of course, it's also relevant for maybe diabetic patients or other people who have diseases complicated by hyperglycemia. So of course, I want to acknowledge my professor, Dr. Trevor A, Mr. Jordan Bird, who helped me with data analysis, the participants who had to undergo that hellish ordeal, and of course, Dr. Carolyn Rickards and Professor Patrice Broussard for organizing. Any questions?
Great job, Anna Maria. We're all giving you our jazz snaps as well, even though our cameras are off. Fantastic job. There's no one in the chat yet, but perhaps while we're waiting for the people to jump in, actually, yeah. Faria Sanjana has asked, how long after the glucose consumption was cerebrovascular reactivity measured?
So we would try and do it pretty quickly. We would measure the glucose response after 30 minutes. And then we would get all of their instruments on, try and get the cerebrovascular activity measured within, I would say, about 5 to 10 minutes. So it's possible that it could go down a little bit, but it's still definitely in that hyperglycemic range for sure.
Didn't we measure before and after the rebreathing test in both cases to see if there were any differences pre-post? And can you remind us what that showed? Yeah,
it was usually only lowered by like a few decimal points. In some participants, they have a really good pancreas, I guess, and the glucose levels would be much lower from start to finish. But usually the participants didn't have a significant difference from before and after their cerebrovascular activity test.
Great. Valerie, I'll just add the reason we chose 30 minutes or so is because of previous study in our lab when we actually tracked every 5 minutes or so during a neuroglucose tolerance test. At the peak on average happened about 30 minutes. So we wanted to try and capture our participants at the peak blood glucose, but we didn't do rebreathing tests throughout that increase and decrease over time, which might be interesting, but this would be about as high as they would have gotten, and that's why we chose that time point. So thanks for your question. I don't see anything else yet. And oh, here we go. Patrice is asking, how would you explain this attenuated CO2 reactivity? Oh, great. The mechanistic question. Do you think acute hyperglycemia would also have a negative impact on CO2 reactivity in response to hypocapnia? That's interesting. So go ahead.
So like I had mentioned before, the reason why we think that the CO2 reactivity is attenuated is because of the nitric oxide. Trevor and I tried to look into insulin as well because we didn't measure insulin for the study. If that's anybody's question, I'm going to answer it now. No, we didn't measure insulin. We didn't have the tools, but we know that insulin increases concomitantly with glucose. But insulin actually increases vasodilation, which might have increased velocity, but we didn't see that. So we're assuming that nitric oxide was the culprit in this case. I'm not sure if there would be a negative impact in response to hypocapnia because that would be a vasoconstrictive response. And I don't know the mechanism for cerebrovascular constriction. I know that nitric oxide does dilation, but I don't know if it would be the same for constriction, if that makes sense. I hope I didn't ramble on too much.
Can you hear me? I never know whether I got this on right or not.
Yeah, you're good.
Okay, yeah, so thanks for that, Patrice. The part of the Duffin hyperoxygen breathing test includes a prior hyperventilation. And that's of particular interest if you're doing respiratory responses as well to identify the ventilatory recruitment threshold. We do have, was it a 1 minute hyperventilation, Anna Maria?
Yeah.
Yeah, so we do have a single point, not convinced they would be at steady state after 1 minute of hyperventilation. So I guess we do have baseline and hypocapnic data, but it wouldn't be the same sort of incremental increase in CO2, or what you're asking for is decrease in CO2 in this case. So not sure if those data are worth looking at, but we're going to think about it though. So thanks for your question. Lots of people are just saying, well done and thanks and awesome job. So congratulations, Anna Maria. You've just given an international talk, so well done.
I was holding my breath the entire time.
Don't forget to breathe. We'll switch speakers now. Thanks, Anna Maria. Wonderful to see your presentation. We're actually going to move Jack up. And so Jack, if you'd like to get on deck and share your screen. Jack is joining us Tuesday evening from Cork, Ireland. I met Jack in 2017 when he approached Ken O'Halloran about doing a master's in human physiology. So we kind of collaborated together and brought Jack over to Canada for a semester of doing some neurovascular coupling training, and then went to Altitude. And you can read Jack's paper in Frontiers of Physiology in 2018, Jack, we published that, looking at neurovascular coupling with Ascent.
Yeah, I think it was 2018. Maybe, yeah, it was. The Ascent was 2017, wasn't it?
Yeah, that's right. Yeah.
So I think 2018.
Jack has become a bit of an expert on neurovascular coupling analysis. And so put your seatbelts on for an analytical journey through neurovascular coupling based upon some data we collected on an expedition to White Mountain last August. So with that, take it away, Jack.
Okay, so Percy, I suppose before I begin, I'd just like to thank Patrice and Caroline for organising this seminar series, and to Trevor A for giving me the esteemed opportunity to present this evening. The focus of today's presentation is basically going to detail the research findings from a research expedition to White Mountain Research Station in Northern California back in August in 2019. The primary physiological variable of interest is neurovascular coupling, which pertains to the coordination between local neuronal activity and regional cerebral blood flow. And primarily within our lab, we've assessed neurovascular coupling through the posterior cerebral artery. The reason being is that through visual stimulation, we can elicit a region-specific increase in metabolic activity of the occipital lobe, an area we know which is perfused by the posterior cerebral artery. The basis of that increase in metabolic demand is driven primarily from the generation and transmission of action potentials on a given neural circuit, which activates a combination of both feed-forward and resultant feedback signaling pathways, which allow for a downstream vascular response. The purpose of that vascular response naturally is to facilitate an increase in the delivery of energy substrates to the metabolically active region. So our lab has kind of adopted a two-step approach for this project in quantifying the NVC response. Step one, we measure and compare the mean and peak velocity achieved during visual stimulation to that of a preceding baseline window. We also measure and compare the total area under the curve achieved during visual stimulation to that of a preceding time-matched baseline window. Step two is, given our understanding of the several, mechanistic pathways involved in NVC and the time-dependent nature of these pathways, we compartmentalized the hemodynamic response, kind of the waveform, into three distinct regions. We have the acute, the mid, and the late response, whereby we measure and compare the magnitude of the hemodynamic response within each of these regions to that of a pre-baseline period. This work was really expanded upon previous research within our group, wherein we measured changes in both arterial blood gases and neurovascular coupling at 4 progressive altitudes during an incremental ascent profile to Everest Base Camp. The primary outcome was that neurovascular coupling response magnitude remained intact. It wasn't until we assessed or looked at the arterial blood draws, which I presented here, which kind of really detail nicely the integrative acclimatization process, where we see significant reductions, or we see the extent of the hypoxic stimulus of the environment with significant reductions in PaO2 immediately upon ascent to high altitude. We see the ventilatory response to that hypoxic stimulus with significant reductions in PaCO2. This, of course, comes as a caveat of an acid-base disturbance, specifically a respiratory-induced alkalosis. We then see examples or evidence of the renally driven compensatory metabolic acidosis with progressive and significant reductions in arterial bicarbonate and reductions in base excess. I suppose the conclusion to this kind of intricate physiological dance is that pH is maintained within normal limits or normal values. And this kind of really got myself and my supervisors talking, Ken and Trevor, that perhaps cerebrovascular function could be impacted by the capacity of pH control or capacity of pH maintenance. And wouldn't it be great to be able to assess or measure and compare the effects of pre-acclimatization on cerebrovascular control against post-acclimatization? And naturally, of course, this would involve a more kind of aggressive ascent profile, which would allow for more rapid ascent, as well as data collection within the narrow window following ventilatory responses to hypoxia, but pre-renal compensation. And this, of course, brought us to turn our attention to White Mountain, which is a research station which facilitates this kind of ascent profile, where we spent 6 days at baseline, so in Mount Royal University, my alma mater, taking baseline measures of NVC and arterial blood gases. We then flew to Las Vegas, where we spent a day seeing the sights, doing all the touristy stuff. We then began our expedition to 3,800 meters, so Barcroft Research Station, where we had a 10-day residency with both measures of NVC and arterial blood gases taken on day 2 and 9. The study methodology was quite straightforward. Neurovascular coupling was measured in response to 5 consecutive visual stimulatory trials. These were initially taken during inspiration of room air. So at Calgary, this was a normoxic stimulus. At altitude, this was a poikilocapnic hypoxic stimulus. Participants were then exposed to a bespoke ventilatory challenge. So at Calgary, this was an isocapnic hypoxic stimulus. And at altitude, this was an isocapnic hyperoxic stimulus. The basis of which was twofold. The isocapnic hypoxic challenge at sea level was designed to emulate the severity of the hypoxic stress that would be experienced during room air at White Mountain. And then vice versa, the isocapnic hyperoxic challenge at altitude was designed to alleviate the hypoxic stress within our participants and return them to an oxygenation status akin to what would be observed during normoxia at sea level. This allowed us to do 3 things. It allowed us, the comparison between normoxia and isocapnic hypoxia at sea level allowed us to assess the independent effects of hypoxia or O2 manipulation on NVC. The comparison between normoxia and the isocapnic hyperoxia on days 2 and 9 at altitude allowed us to really isolate the effects of hypocapnia or CO2 manipulation on NVC. And then the comparison between normoxia and room air on days 2 and 9, so poikilocapnic hypoxia at altitude, allowed us to assess the combined effects of hypoxia and hypocapnia. We also assumed that due to the rapid ascent profile or nature of the ascent profile within this expedition, that the measurements on isocapnic hyperoxia on day 2 and poikilocapnic hypoxia on day 2 would be in the backdrop of respiratory-induced alkalosis, which would be normalized by day 9. So the aim of the study was to investigate the effects of acute high altitude exposure and NVC response magnitude. We also tried to investigate the independent and combined effects of arterial blood gas manipulation on NVC response magnitude. And we hypothesized that the respiratory-induced alkalosis following acute exposure to high altitude would impair NVC response. Aside from the kind of traditional cardiorespiratory measures that you would routinely observe during altitude exposure, there's 3 key measures that I wanted to present to you, which kind of demonstrates, I suppose, the success of our methodology. Here we look at changes in SpO2, end-tidal O2, and CO2 for each of our locations, for each of our gas challenges. And there's 3 kind of key observations from this slide or from this table. Firstly, the hypoxic stimulus at Calgary was both successful in incurring a hypoxic stimulus, but the magnitude of that hypoxic stimulus was directly comparable or emulated really nicely the extent or severity of the stimulus that was seen at both White Mountain on days 2 and 9. Secondly, that the hyperoxic gas challenge at altitude was successful not only in alleviating the hypoxic stress, but also returning participants to an oxygenation status akin to what was observed during normoxia at Calgary. And then finally, aside from the time-dependent reductions in end-tidal CO2, which you routinely see, when we make within location comparisons, we can see that end-tidal CO2 was extremely well controlled for, which, you know, is always important when you're doing isocapnic testing, that you control for end-tidal CO2 or CO2. Moving on then, one thing we try to really push in our lab, and maybe I picked it up from spending too long with Trevor and Ken, is this idea of controlling, and particularly in controlling for secondary variables which might influence your primary variable of interest. And at its core, at its basis, neurovascular coupling is essentially just a cerebral hemodynamic response to a stimulus. And it's then important that we control for other variables which also affect cerebral hemodynamics. And in particular, within the literature, we know that the cerebral blood flow is intrinsically linked to the cardiorespiratory system. And so what we've done here is we plotted the relationship between cerebral velocity in our middle cerebral and posterior cerebral artery with that of minute ventilation, mean arterial pressure, and heart rate. And we can see quite a linear relationship in both vessels. It's therefore kind of crucial or imperative that when we're kind of making assessments of hemodynamic responses between a baseline period and a visual stimulatory period, that we control for each of these parameters. And that's kind of what we've done here. So here we have Calgary, White Mountain day 2, day 9. We have each respective gas challenge at each location and each parameter, so minute ventilation, heart rate, mean arterial pressure. And along the y-axis here, what we're doing is representing the percentage change in each of these parameters between our baseline period and during visual stimulation. And we can see a kind of consistency for each gas challenge and for each altitude location, that each of these variables remain remarkably stable between baseline and visual stimulus. This gives us kind of confidence that any changes or any hemodynamic response we see during visual stimulation is not a consequence of changes in our cardiorespiratory system. The next thing we like to look at, again, more control, is the kind of more assurance of correct vessel insination. And the easiest way to do that, in my opinion, is the inclusion of a negative control. So here what I've done is I've presented the group averaged hemodynamic response to visual stimulus in both the middle cerebral artery, which perfuses the anterior region, and the posterior cerebral artery, which perfuses the posterior region, or the occipital lobe, in response to visual stimulus. For each gas challenge at each location, so Calgary, I'm at day 2, day 9. And just from visually looking at these graphs, you can see there's aberrant differences in the hemodynamic waveform during visual stimulation. And when we graph that then, when we look at Calgary, White Mountain day 2, day 9, we have each of our gas challenges along the x-axis. We have delta in cerebral velocity as both a measurement of delta and also relative change or percentage change from baseline. And we can see a kind of consistency in that. For each gas challenge at each location, there was a superior hemodynamic response to visual stimulation relative to middle cerebral artery. So what we've kind of concluded from this is that there is evidence of a hemodynamic response, and that hemodynamic response is not a consequence of any changes in our cardiorespiratory system or external variables which influence cerebral hemodynamics. So therefore, it must be a consequence of downstream neurovascular signaling. So when we take a closer look then at the cerebral hemodynamic response magnitude through the posterior cerebral artery, once again, we presented each location, each gas challenge, we presented the average waveform. What we can see is that although there are shifts in the baseline, so the starting point, expected with changes in gas challenge, we see that the overall shape and magnitude of each of these responses is quite similar. And when we present that here, or graph that here, we have each of our 3 neurovascular coupling response parameters. We have the mean and peak change in velocity, we have total area under the curve change, and we represent those both as a delta and as a percentage change or relative change from baseline. And what we can see is that at each altitude for each gas challenge, that the overall magnitude of the hemodynamic response to visual stimulation is intact, and inferring that NVC is in fact intact with acute and prolonged exposure at high altitude. Next then, when we looked at the independent combined effects of arterial blood gas manipulation, what we've done is here we presented changes in both SpO2 and end-tidal CO2 for each specific gas challenges, which are grouped dependent upon the type of stimulus we wanted to look at. So here we have the independent effects of hypoxia, hypocapnia, and a combination of the two. We have the corresponding neurovascular coupling response magnitude for each of our parameters, so the mean change in velocity, the peak change in velocity, the area under the curve change as a percentage change. And what we can see is that despite aberrant differences in arterial blood gases, that neurovascular coupling response magnitude remains intact. So overall conclusion, neither isocapnic hypoxia, isoxic hypocapnia, nor poikilocapnic hypoxia have any effect on NVC response magnitude. After this, our kind of final analysis metric, we wanted to look at region-specific differences in the hemodynamic response, and whether or not there were any changes in that region-specific hemodynamic response with changes in blood gases or prolonged exposure to high altitude. So here we have the acute response, the mid response, and the late response as both percentage change from baseline and deltas at each of our 3 locations for each gas challenge. And what we can see once again is that overall regional the hemodynamics of different regions were intact following prolonged exposure to high altitude across each of our different gas challenges. While this might be a bit speculative, we do think that this indicates that both feed forward and feedback mechanisms involved in MVC remain intact. So if there was, I suppose, a kind of a figure which concludes the narrative of this project, it would be this, where we presented the group averaged MVC waveform for each gas challenge at each location. And we can see that despite shifts in baseline PCAV, that the overall shape and magnitude of the neurovascular coupling response This naturally, of course, goes against our hypothesis, as we imagined or we envisioned that the acute exposure to high altitude would have impeded neurovascular coupling response magnitude. But perhaps similar to what we observed in altitude, that the truth lies in the blood gases. So here again, we have the extent of the hypoxic nature of the environment with significant reductions in PaO2. We see not only elements of the acute ventilatory response to hypoxia with reductions in PaCO2 on day 2 at White Mountain, but also evidence of the ventilatory acclimatization to hypoxia, or an increase in ventilatory loop gain with progressive reductions in PaCO2 with time spent at altitude, despite similar PaO2 stimulus. Indicative that for a given PaO2 stimulus, we now get a greater ventilatory output. We see evidence of the acute response, renal compensatory response to that respiratory alkalosis with progressive and stepwise reductions in arterial bicarbonate as well as base excess. And I suppose what we weren't anticipating was that pH would be normalized. And the reason we weren't kind of anticipating this was given the scope or the context of the severity of the stimulus. Just for a bit of context, the delta in the altitude that we achieved on this expedition was approximately a transition from 600 meters of Vegas to 3,800 meters of Barcroft. That's a delta of about 3,200 meters across a 6 to 8 hour period. And to put that into context, that Nepal, to achieve that similar delta in altitude, it took us about 8 days. So what this demonstrates first and foremost is the remarkable capacity and time frame in which the integrative physiological acclimatization process can occur. Which kind of caught us by surprise. So it appears that, you know, the kidney is more than capable to adapt to the severity of high altitude exposure. Next, then we just see the evidence of the chronic acclimatization of the renal system with erythropoietin signaling in significant increases in hemoglobin and resulting hematocrit by day 9 at altitude. So our conclusion and take-home messages from this is that MVC response magnitude is unaffected during acute high altitude exposure and isohydric manipulation of arterial blood gases. This builds on previous findings by our group, whereby we now know that neurovascular coupling response remains intact despite acute or incremental exposure to high altitude. It's important that we discuss that we point out that both these observations remain in the background of a normalized pH. Naturally, I'd like to acknowledge everybody who's involved in, I suppose, the data collection process of this study. For anybody who's been to kind of these high altitude expeditions, you'll know that it often takes a village to get these studies over the line. There's a lot of moving parts, a lot of spinning plates. And I'd like to thank everybody who's involved either as a participant or in the data collection process. And thanks for listening. And I'll answer any questions you might have.
Thanks so much, Jack. We're all jazz snapping for you.
I hope I wasn't too far over in time, was I?
It's all good. We'll go a few minutes over the seminar if everyone's willing to stick around to see Taylor's wrap up of this whole story. But I think we should probably make time for a bit of questions because we're looking for feedback as we package this for submission. But great job in eliciting a neurovascular coupling response in us, Jack, when we're looking at your dizzying analysis. I think the spinning plates analogy is my favorite one that you've come up with respect to integrative physiology at altitude. So any questions for Jack or any comments or feedback on our findings? Rehan, sorry, I can't pronounce your last name, Juneo. Great work. What is the specific visual stimulus for MVC? And may be missed, but what sex and gender breakdown?
So the visual stimulus we use is one we've used in the lab previously, and when we published it previously, where we use a strobe light stimulus that has a set intensity of 6 hertz. It's quite robust and it's quite repeatable. And the sex gender breakdown, from what I remember, was about half and half. But we do have unpublished work at the minute that we're kind of waiting to get out, which demonstrates very little, if not no, gender effect on neurovascular coupling response to this stimulus anyway, to this 6 hertz visual stimulus. So that gives us confidence that gender was not a covariate in our analysis.
Thanks for your question, Rianne. Yeah, I look forward to seeing that sex difference or the lack thereof come out. AJ is here. Nice to see you, AJ. Thanks for a great talk, Jack. How do you account for the possible effects of hypoxic vasodilation on your NVC results at high altitude?
Yeah, that's one of the caveats, unfortunately, of Doppler, is that it doesn't provide you an index of changes in diameter. Now we know that hypoxia causes vasodilation of the cerebral arteries, so it's one of those. I suppose I don't like, unless you're going to be doing a duplex ultrasound, it's very hard to control for it with Doppler ultrasound. I suppose I don't know. Let me refer on it. Let me think. We don't. I suppose. Yeah, I suppose the take-home message is we haven't really accounted for hypoxic vasodilation on our MVC results. We try to control for it as best we can with changes in ventilation, or standardize it with changes in ventilation and cardiovascular driving forces. But one thing we don't get to look at, we don't get to index it against the basal tone of the vessel or the basal diameter of the vessel at each testing point, unfortunately. But next time.
Yeah, it's tricky. Thanks, AJ. Yeah, it's tricky because it's difficult to hold a continuous IC or VA with duplex ultrasound for the length of the neurovascular coupling protocol that we talk about. But the light stimulus probably just isolates downstream arterioles. But you're right that some evidence is showing that the conduits might also dilate in hypoxia as well. So I guess that's what he's getting at. Yeah, caveats for every method, right? Yeah. Thanks, AJ. Don't see anyone else now, so perhaps please go ahead and email us if you have any feedback for us, because we're looking forward to getting this out soon. Wonderful job, Jack, and really comprehensive analysis. And if I ever have to see you waving a finger in front of my face again, I'll probably snap. So, but incredible amount of analysis on that. So well done. Thank you. We'll just wrap up the story then. After Jack's preliminary analysis, we saw that even within 24 hours that people's pH was returned to baseline values. And so they weren't alkalotic anymore as we anticipated, because we built that into the study design. But the kidneys were so good, as Jack said, that we got back to baseline values, and that may have contributed to the lack of result or the stability we observed. And so coming back to Calgary last fall, we decided to make an amendment to our ethics to just investigate the acute effects of respiratory alkalosis and NVC. And Taylor Bader, who just finished his BSc in health science at Mount Royal, was in that advanced class and took some NVC training. And we decided as part of an independent project course for him to collect a bunch more data on this particular question. So Taylor has completed his BSc now and has started a master's degree at the University of Calgary, and is taking time out of his schedule to come back and present the undergraduate project to us. So thanks, Taylor, for your efforts, and look forward to your talk. I can see your slides just fine, so start when you like.
Perfect. Okay. Well, hi, everyone. Let's wrap this up. Today, I'm talking about the effects of acute hypocapnia on neurovascular coupling magnitude. Of course, a big thanks to my colleagues at Mount Royal University, University College Cork, and then, of course, everyone being here today. So a little bit of background. As everyone else has mentioned, 20% of the resting oxygen intake you breathe in is consumed by your brain. And this is pretty substantial, being that it's only 2 or 3% of your body weight. On top of this, it's also not storing many nutrients, and being so metabolically active, it's producing a lot of metabolic waste. To overcome these, it requires constant as well as highly regulated blood supply to also get it through a variety of different scenarios. Examples could include if you start using your muscles, this can't take away the blood from going to your brain, as well as changes in cerebral activity. Sometimes you're just blankly staring at your computer screen, and other times you're actually using your brain and thinking about a lot of complex things. Again, you have to adapt to these things. Now, there are quite a few different mechanisms that maintain this, but today I'm going to be talking about 2 of them. The first is cerebrovascular reactivity, or CVR. And this is how the blood vessels change based on the blood gases within them. For our study, we're specifically looking at CO2 and O2. Additionally, we're looking at neurovascular coupling, or NVC, which Jack was just talking about. And again, this is the regional matching of blood flow based on metabolic demand, where the neurons and the astrocytes are working together to have the end feet change the amount of blood flow going through that area. Now, a little bit more on cerebrovascular reactivity. If you were to have a decreased amount of CO2, something called hypocapnia, this can be just from hyperventilating. This would cause vasoconstriction through increasing the resistance in the blood vessel, decreasing flow. Now, on the opposite end, if you were to decrease O2, hypoxia, this would cause vasodilation, reducing the amount of resistance and increasing flow. Now, a little bit more background on neurovascular coupling. So again, this is a regional matching of blood flow based on neuronal activity. Commonly, this is monitored through the posterior cerebral artery, which is perfusing the blue area here, primarily the occipital lobe, as well as some of the temporal. Now, this is commonly looked at because it's easy to change the activity in that area. If you open your eyes, you increase the neuronal activity because now you're processing images. And by doing this, you're also increasing the blood flow going through that vessel. Additionally, if you close your eyes, it's a new way to decrease neural activity in that area, which would also decrease the blood flow. Now, in studying this, another common method is the transcranial Doppler ultrasound, similar to previous studies we were just talking about. With this, you can measure the blood velocity going through that posterior cerebral artery, where here we just have a raw tracing from one of our subjects. Now, these rapid changes where the gray areas are their eyes closed and the white areas are when their eyes are open are the result of them opening and closing their eyes, where you see this sharp increase in blood flow going through that vessel versus when their eyes are closed. Now, this dramatic change in blood velocity going through that blood vessel is the neurovascular coupling response. So how do these two different mechanisms affect one another? Well, a study back in 2011, Szabo and colleagues found that neurovascular coupling magnitude was attenuated during acute hypocapnic conditions. They measured this in a laboratory setting where things are pretty controlled. They only measured 2 levels of CO2. So they had a baseline and then they had heavy hyperventilation. And additionally, they only looked at the peak response, which I'll get more to in a minute here. Now, on the opposite end, as Jack was just talking about, neurovascular coupling magnitude is unchanged when individuals go to altitude. Now, these are different conditions. They are experiencing chronic exposure where there's renal compensation, helping to maintain that blood's pH, as well as they're experiencing graded altitude increases, where it's not just, although it would probably be convenient getting dropped off at the Everest base camp, they're slowly working their way up. And lastly, they're experiencing these conflicting stimuli of both hypoxia and hypocapnia. So what we wanted to do is add a little bit more to this and test controlled and graded hypocapnic steps, similar to what they experience at altitude, but without that conflicting stimuli of hypoxia. Additionally, we hypothesized that there would be a variable decrease in neurovascular coupling magnitude during these acute hypocapnic steps. So the amount of neurovascular coupling response would decrease at each step. Measuring this, looking at the 2 primary ones, we were measuring their partial pressure and tidal CO2. Using a gas analyzer off to the side, we were measuring as the individuals were exhaling. Additionally, we were using the transcranial Doppler ultrasound, again measuring that change in blood flow going through the posterior cerebral artery. Our study had 3 different sections to it. The first is normal breathing. So this is no change in their partial pressure and tidal CO2. So past this, we had 2 levels of coached breathing, where we had a mild hyperventilating, -5 CO2. This would be similar to just going up a flight of stairs. And then we had a heavy hyperventilation, -10. And this was a bit more severe, where they're at -10 partial pressure and tidal CO2. Now, during each of these different breathing interventions, we had the same neurovascular coupling intervention, where we had a prolonged baseline of their eyes being closed, followed by 5 minutes of our visual stimulus, where we had their eyes closed for 30 seconds, open for 30 seconds, close for 30 seconds, open for 30 seconds, and so on. Where similar to Jack's study, we were shining a strobe light in their eye when their eyes were open. Now, our results from running this protocol. In total, we had 17 participants in our analysis. Now, when bringing down their CO2, again, this is through making them hyperventilate, we aim for plus or minus 1 from what their baseline was. So being here in Calgary, we started around 36.7 on average, brought them down to 31.6, and then brought them down further down to 26. Something else to note on some of these secondary stats is that we also saw a decrease in the baseline of this, the blood velocity going through the posterior cerebral artery, where at rest it was closer to 40, while individuals at -10 were closer to 30. Now, our neurovascular coupling response analysis. We measured it in 3 different ways. First of which, we were looking at peak. So similar raw tracing from before. We just gathered a 1-minute baseline while their eyes were closed, and then compare this to the max peak during each of the 30 seconds that their eyes were open, averaging these 5 together. From this, we found from 0 to -5, again, that mild hyperventilation to heavy hyperventilation, that there was a statistical significant decrease, both at -5 and -10 compared to 0. Where here we see the change in blood flow going through that posterior cerebral artery decreasing slightly. Next, we wanted to measure the mean. So with this, similar to peak, we took a 1-minute baseline while their eyes were closed, but then compared it to the entire 30 seconds that their eyes were open. Again, averaging 5 stimuli together. From this, we saw no statistical difference between 0, -5, and -10. We're a similar structured graph here, where again, we're seeing the change in blood flow going through that posterior cerebral artery. And lastly, we wanted to look at area under the curve, since some of this data can be kind of spiky. From this, we compared the 30 seconds while their eyes were closed to 30 seconds, again, all of the interventions together averaged. And from this, we found, again, no statistical difference between 0, -5, and -10, where here we're looking at the, again, area under the curve for each of these. So what can we get from all this? Well, this has been one of very few studies that have actually shown a change in neurovascular coupling response, specifically to the peak here, where Savo and colleagues back in 2011 were looking at 0 and actually past -10. I think their average was closer to -12, partial pressure and tidal CO2. We found that there was an earlier plateau in this neurovascular coupling attenuation, where we saw similar results at -5 and -10. Now, with all that being said, there was no change in both the mean and area under the curve neurovascular coupling response. And being that the peak change was so small, it's hard to say if there's actual any physiological changes going on here. So overall, it's probably pretty safe to say that neurovascular coupling is, again, a pretty robust brain blood flow mechanism. And despite these individuals hyperventilating down to -10 partial pressure and tidal CO2, most of these metrics were maintained. And with that, I'd just like to wrap it up. Give a big thank you to, again, my colleagues at Mount Royal University, colleagues at Mount Royal University College Cork, and then Dr. Caroline Rickards and Dr. Patrice Bessard for putting all of this together. It's really great being part of this larger community. I'll have to open up to questions.
Well done, Taylor. Congratulations, and thanks for that, and for wrapping up the story of neurovascular coupling in various contexts of hypocapnia. Ryan Hoyland, who world expert on this area, has asked a question in the chat. In your methodological image, the participant was sitting, whereas many other studies were conducted supine. Out of curiosity, do you have any data comparing NVC in these 2 different body positions?
That's an interesting question, and I've actually, I haven't looked at that before. Most of it was trying to get the participants comfortable, and sitting was pretty easy for that when looking at MAP. I wouldn't want to quote it, but it was never brought up to my attention that there are differences between supine and sitting. So I'd feel pretty confident that the results would be similar, but that's something to consider for sure.
Good question, Ryan. all the studies in MVC we've done have been in sitting position. So Jack's 2018 Frontiers paper with Ascent, the data Jack just showed, and then the data Taylor just showed were all in sitting position. We haven't tried it supine or compared it. I can tell you that cerebrovascular reactivity is unchanged with body position. Mike Timko and I did a study years ago where we hung people upside down at 90 degrees head down tilt, and cerebrovascular reactivity during hyperoxygen breathing was unchanged. So I'm not sure small changes from sitting to lying down would make a difference, but maybe. It's hard to know. Apparently I've asked a question to everyone. What is the physiological relevance to AUC for a velocity waveform? It is by definition a position vector from a calculus perspective. So this person's asking what the significance of the area under the curve analysis might be, and I guess we can leave that for Taylor and or Jack to address.
Yeah, I'll just try and pull up a graph here. So when looking at this raw tracing is actually standardized, I believe, to, I think it's half a second. I'd have to confirm with Trevor with that. But the data itself is even more spiky, actually. So by looking at area under the curve versus looking at mean, you account for some of these just random spikes in the response, and it helps really show you a true mean versus where the mean might show some random fluctuations that aren't supposed to be there. I guess it cleans up the data a bit. I'm sure Jack could answer that a little bit better.
Jack, do you want to jump in just because you've pioneered a lot of this analysis in the last couple of years?
Yeah, so we've talked about this a good bit, Trevor, haven't we, about trying to identify what are the kind of the most important parameters in neurovascular coupling analysis. And what is kind of, I suppose, what we've noticed now is that area under the curve and mean tend to correlate well with each other in terms of the magnitude of response. I like to use area under the curve just because it gives, I suppose, a more comprehensive analysis of the overall waveform or the overall change in hemodynamic response during the entirety of the visual stimulus in comparison to just picking out the peak. And as Taylor alluded to there as well, the mean kind of can be sometimes difficult, particularly in participants where you do get a lot of noise. I like to look at area under the curve because that noise doesn't take up such a larger, I suppose, area, rather than whereas the mean velocity achieved during that noise or that spike can greatly influence the average velocity achieved during visual stimulation. So it's a difficult one to tease out. We've looked at it a couple of times. I think the understanding is that they too can tend to follow each other in that both mean response and area under the curve response tend to show the same thing both times in terms of magnitude of response. I just like including area under the curve because it gets rid of the, I suppose, the confounding effect of any spikes affecting mean velocity or any kind of noise.
Cool. Thanks, Jack. Yeah, I think Ben Stacey's got some data that we saw last fall at the conference in the UK that we look forward to seeing that have to do with altitude as well. Any other questions for Taylor? Ryan's asking again, given there isn't a graded response of MVC to changes in entitled CO2, do you think it's CO2 per se, or rather an increase in cerebrovascular tone prior to the visual stimulus that matters?
Yeah, that's a good question. And something that we were trying to control with that was randomizing the order. So we actually had individuals start at 0, and then sometimes we had them go to -5, then to -10. And other times we were going from 0 to -10 to -5. And it did seem as individuals were sitting in the chair for longer periods of time, the data did go down slightly, but we did try to control this by randomizing the order of it. But yeah, I would say there are quite a few different mechanisms overall at play affecting this.
I think is what he's getting at is, as you showed in your baseline table, that hypocapnia causes a relative vasoconstriction. So the starting point is a constricted vessel compared to baseline. He's following up and saying that, for example, increases in vascular tone with indomethacin, which is a drug that reduces brain blood flow, and hypocapnia together have shown similar influences on dynamic cerebral autoregulation, another metric of cerebral vascular regulation, indicating it was non-specific influence of basal tone. So it's an interesting thought that the background tone from the hypocapnia might have caused a slight reduction in the peak. But as Taylor pointed out, basically it's, even though it's physiologically reduced, or sorry, statistically reduced, as Taylor pointed out, I'm not convinced it's physiologically meaningful because it's only a couple of centimeters a second difference. So cool. We'll think about some of that in the writing in the manuscript, Brian. Thank you. Last chance for questions for Taylor, and then we'll let you all go. We've lost a few participants, but there's still some people sticking around. So, Karen, it looks like I'll turn it back over to you because I understand you have a wrap up. So thanks so much to Patrice and Carolyn for organizing